In the realm of genetics and evolutionary biology, few topics have sparked as much intrigue and controversy as the idea of a humanzee, a hybrid of a human and a chimpanzee. This concept, which straddles the line between science fiction and scientific possibility, was thrust into the spotlight by Gordon Gallup, an evolutionary psychologist best known for his work on self-awareness in animals. Gallup's startling claim that a humanzee was born in a US laboratory in the 1920s challenges our understanding of species boundaries and raises profound ethical questions. In a 2018 interview, Gordon Gallup, a professor at the University at Albany, made the sensational claim that a human-chimpanzee hybrid was successfully created in a US laboratory nearly a century ago. According to Gallup, a former professor confided in him that the experiment took place in the 1920s at a research facility in Orange Park, Florida. A female chimpanzee was artificially inseminated, resulting in a pregnancy and the birth of a live offspring. Gallup stated that the infant, which exhibited both human and chimpanzee characteristics, was put down by the researchers due to ethical and moral concerns. The early 20th century was a period of burgeoning interest in genetics and evolutionary biology. The rediscovery of Gregor Mendel's work on heredity in the early 1900s paved the way for a deeper understanding of genetic principles. During this time, scientists were increasingly interested in the genetic similarities between humans and other primates, particularly chimpanzees, our closest living relatives. The idea of creating hybrids to study genetic and evolutionary questions, although controversial, was not entirely outside the realm of scientific inquiry. Attempts to create hybrids between different species were not unheard of. In the early 1920s, Soviet biologist Ilya Ivanovich Ivanov conducted experiments to create human-ape hybrids, although none were successful. Ivanov's work, though ultimately unsuccessful, demonstrated a scientific interest in exploring the boundaries of hybridization and the genetic relationship between humans and other primates. Humans and chimpanzees share approximately 99% of their DNA, suggesting a high degree of genetic compatibility. This similarity has led some scientists to speculate that interbreeding between the two species could be possible, at least theoretically. The chromosomes of humans and chimpanzees are sufficiently similar that fertilization and the initial stages of embryonic development might occur. Despite genetic similarities, significant biological and ethical obstacles make the creation of a humanzee highly improbable. Differences in chromosome number and structure between humans and chimpanzees pose challenges to successful hybridization. Additionally, the gestation and development of a hybrid embryo would require overcoming substantial physiological and immunological barriers. Even if a hybrid were born, it would likely face severe developmental and health issues. The creation of a humanzee raises profound ethical questions. The deliberate hybridization of humans and chimpanzees challenges fundamental notions of human dignity, identity, and the integrity of species boundaries. The potential suffering and exploitation of both human and non-human participants in such experiments are significant ethical concerns. The idea of creating a being that is neither fully human nor fully chimpanzee poses moral dilemmas about the rights and treatment of such a creature. Scientists have a responsibility to conduct research that respects ethical standards and the well-being of all subjects involved. The alleged expiration of the humanzee infant, as described by Gallup, underscores the complexities and potential consequences of such experiments. The scientific community must navigate the balance between curiosity-driven research and ethical considerations, ensuring that the pursuit of knowledge does not compromise moral principles. The concept of a humanzee, if proven feasible, could provide unique insights into human evolution, genetics, and the nature of species boundaries. Studying such a hybrid could reveal aspects of our genetic heritage and evolutionary history that are otherwise inaccessible. However, the moral challenges associated with creating and studying a humanzee may outweigh the potential scientific benefits. In a pioneering experiment, scientists have successfully generated the initial human-monkey chimera. The study details the development of the initial embryo that consisted of a combination of human and monkey cells, which was cultivated for a period of 20 days. Under the leadership of Juan Carlos Ispizua Belmonte, this study represents the result of extensive research over several decades on the early development of embryos in non-human animals. Belmonte aims to apply this knowledge to humans. However, this development is certain to provoke significant ethical concerns regarding the consequences of merging human cells with those from a distinct species, even if it is a closely related one. The report was accompanied by input from ethicists, who provided their analysis on how the research should be understood and recommended cautious measures for further exploration in this field of study. Respected for his work in embryo development, Belmonte is a professor in the Salk Institute for Biological Studies Gene Expression Department. He is extremely explicit about his motivations for doing the experiment and the direction he thinks it will go. During conversations with news outlets during the progression of the experiment since 2019, he provided a meticulous explanation of the biological mysteries he aimed to unravel and the deficiencies in our understanding of early development that he sought to address. According to him, the initial stages of human embryo development are the most mysterious aspect of human growth. Although we possess considerable knowledge about postnatal development and some understanding of prenatal development, our understanding of human development during the initial 2 or 3 weeks following fertilization is extremely limited. All our knowledge of early embryogenesis is derived from many laboratory animals, such as rodents, mice, and worms. However, our understanding of this process in humans remains extremely limited. Belmonte's Cell publication details the successful creation of a human-monkey chimera capable of sustaining life for a duration of 20 days. This time frame allows for the thorough examination of the crucial initial phases of development. More precisely, he and his team were investigating the distinct signals emitted by the early-stage cells that trigger the transformation of a single fertilized cell into the complex network of millions of cells, tissues, and organs that form a human body. The strong evolutionary relationship between primates and humans enables this possibility. Belmonte had conducted comparable studies with human and pig embryos, but was unsuccessful. Belmonte intends to acquire knowledge about the early human embryo development by utilizing the human-monkey model. This understanding will aid in comprehending diseases and potentially producing human tissues for organ transplantation. To avoid ethical concerns, Belmonte aims to conduct experiments on a species like the pig, which is evolutionarily further from humans and more convenient for experimentation. The issue arose when attempting to culture and nurture embryos in non-human primates, who are the animals most closely related to humans on the evolutionary scale. These monkeys exhibited a level of complexity similar to that of humans in this regard. The initial challenge was related to the lack of sufficient financial resources. The National Institutes of Health, which is the primary provider of financing for fundamental biomedical research in the United States, does not provide financial support for studies involving human chimeras. Belmonte sought support from his home country of Spain, where this type of work is permitted, as well as from private foundations and the state-funded California Institute for Regenerative Medicine. Subsequently, the biological obstacles followed. Prior to 2019, there had been no instances of non-human primate embryos being grown for the duration of about 3 weeks required for them to reach the gastrulation stage, which was of particular interest to Belmonte for further comprehension. In that particular year, Belmonte joined forces with colleagues, under the leadership of Wei Jiji, at the Primate Biomedical Research Facility in China. Together, they achieved the successful cultivation of monkey embryos for a duration of 20 days. This milestone was reached precisely during the crucial stages of gastrulation, when significant developmental changes begin to occur. The study had a minimal impact on the broader world, as it was quickly overshadowed by the events of 2019. However, the progress was crucial for Belmonte's ambitions to create chimeras. By utilizing a monkey embryo capable of surviving up to 20 days, researchers may initiate the introduction of human cells into the embryo. This would allow them to investigate the initial series of genetic, molecular, and chemical transformations that govern early development. The long-awaited study findings provoke enthusiastic delight, even from this reserved scientist. Belmonte observed the extraordinary fusion of monkey cells and human cells that he had inserted into a developing monkey embryo. The human cells were derived from a stem cell line in China, utilizing Yamanaka's technique of reverting an adult cell to its embryonic form by reprogramming. A total of over 3,000 reprogrammed human cells were inserted into over 130 monkey embryos, with each embryo receiving 25 cells. As time passed, the number of viable embryos decreased, and on the 19th day, only 3 embryos were still viable. Belmonte acknowledges that the initial attempt was not a resounding success. However, he is cautious about determining the efficiency of the process definitively, as this study only involved a single injection of human cells at a specific time. Previous experiments with mouse and rat cells included varying numbers of cells introduced at different time points during embryonic development. However, the observation that these cells effectively communicate and mature in a rather typical manner strongly indicates a high level of efficiency. This initial attempt at creating human primate chimeras does demonstrate that cells from different species may communicate with one another. This indicates that Belmonte's idea of creating a model outside of the body to study the early phases of human development may well be achievable. By identifying the signals and processes that human cells utilize to differentiate into various tissues and organs, he can replicate this environment in pig embryos. This will ultimately enable the regeneration of human tissues, such as skin grafts for burn patients, as well as heart, lung, or liver tissue to replace damaged and diseased cells. Belmonte is currently engaging in collaboration with experts and pig farmers in his home country of Spain to establish the subsequent research endeavors. He is fully cognizant of the fact that his work is contentious and would be perceived by some as contravening the rules of nature. Prior to commencing his research on human-monkey chimeras, the scientist sought guidance from 3 impartial ethicists and acquired consent from the institutional review boards responsible for overseeing experiments involving human subjects or tissues. These approvals were received from both the Salk Institute and the Collaborating Institute in China. Hank Greeley, the head of the Center for Law and the Biosciences at Stanford University, expressed his approval of the experiment, stating that he does not have any significant objections to it. This is because the embryo will not be implanted for further gestation. However, he acknowledges that doing such tests might cause concern among individuals and may lead to misconceptions if not adequately clarified. Greeley highlights a potential worry with consent for these studies. He emphasizes the importance of ensuring that cell donors are provided with comprehensive information regarding the possibility of their cells being utilized for chimera research. The majority of informed consent forms address the conventional studies that may involve the use of these cells. However, they may not be suitable for more advanced uses like this one, as they were not anticipated at the time the cell lines were given and created. The human cells utilized in Belmonte's research are derived from a stem cell lineage that was established and sanctioned by the Ethical Committee at Peking University. These cells were previously employed in studies conducted by Belmonte and his colleagues in China, which served as a foundation for their current work. Although the consent and study were accepted by the review boards of both universities, it remains uncertain if the consent explicitly addressed the chimera research. In addition to that concern, he asserts that individuals may not share the same level of ethical acceptance as he does regarding the scientific merits of the study. The scientists stated that the public becomes most concerned when something unexpected happens that has alarming implications for which they were not prepared.